Okay. Wait, I want to see this. Wait, I can just look it up. Oh, I see it.
Version ends in. So angiotensinogen is inactive. Angiotensin is active. Angiotensin 1 is further converted to an even more potent variety, called angiotensin 2, by a very intelligently named enzyme, the angiotensin converting enzyme. Angiotensin converting enzyme converts angiotensin 1 into angiotensin 2. And angiotensin 2 then has two direct effects. The first direct effect is that it is a very powerful systemic vasoconstrictor, and the second direct effect is that it helps to increase aldosterone release. So let's look at each of the effects independently and how they would affect blood pressure. So, systemic vasoconstriction. This means all of the arteries in the body will be constricted or narrowed. This effectively makes the container in your body for blood smaller. It may have not really changed the volume of the container. You just made it smaller, so the pressure is going to go up. And if you make the container smaller, it makes the container in your body for blood smaller. It may have not really changed the volume of the container. You just made it smaller, so the pressure is going to go up. And if you make the container smaller, that's going to increase pressure in the container. And that was our initial problem to start with, right? Our blood pressure was falling. The aldosterone is going to increase sodium reabsorption at the distal tubule. So if more sodium is being reabsorbed into the blood, then blood osmolarity is increasing. And if blood osmolarity increases, that's going to trigger a release of antidiuretic hormone, or ADH. ADH, recall, is going to allow us to retain more water to help balance out that increase in osmolarity. If you're very, very salty, then you're going to want to take some water out of the urine to help balance out that salt. So this is going to increase water absorption. That's going to increase blood volume. And now we've packed more about stuff into the container. So that's going to increase blood pressure. So not only have we made the container smaller by vasoconstriction, the fact that we're pushing more blood to the kidney will also help increase filtration rate at the kidney. But remember, this is certainly going to fix the systemic problem. If we increase the pressure overall, we're going to be perfusing our organs better. And the simple fact that we're pushing more blood to the kidney will also help increase filtration rate at the kidney. But remember our glomerulus. Let's just review our nephron we had, the glomerulus here in the capsule and all the tubule coming after. And we said that the glomerulus was unique because it had an artery in and an artery out. And this would help regulate flow into and out of the glomerulus and helped to regulate pressure. So if angiotensin 2 is acting to systemically vasoconstrict all of the arteries in the body, then both of these vessels are going to be constricted. And that's fine for the efferent arteriole. We want to let less blood out of the system, but not so good on the afferent side because that's going to let less blood in. So less blood in, less blood out means we haven't really changed pressure inside the glomerulus here. So here's our last player in blood pressure regulation, and that's a structure called the juxtaglomerular apparatus, or the JGA. The juxtaglomerular apparatus is just a contact point between the artery and the distal convoluted tubule. Now, the way we have our nephrons drawn, it doesn't really look like they would ever contact one another, but they are actually far more twisted. And they do in fact come back and contact each other. So the afferent artery, the cells in the artery, there are baroreceptors. So they are monitoring blood pressure. And if blood pressure falls, these are the cells that secrete renin. And renin sets off that whole pathway we just looked at with angiotensinogen becoming ultimately angiotensin 2, and the effects from that. The cells on the distal tubule side of the JGA are chemoreceptors, and they are monitoring concentration, or osmolarity of the filter.
Bro. Wait, who is the other one? Oh, she is Riley. She is Riley. She's literally from Israel. Gal Gadot, like she's literally Jewish. Oh, she's like the Wonder Woman girl. Oh, so I didn't like her because she's Jewish or because she supported Israel?
Filtrate in the distal tubule. And if they notice that osmolarity has fallen, filtrate osmolarity has gone down, the fluid is more dilute, then these guys are going to do two things. First, they're going to stimulate the artery to release renin, really just to make sure that the system has been kicked into gear. And second, they're going to directly dilate the afferent artery. Now, easy enough to do because they're in contact with one another, so the distal tubule can just dump some little vasodilator substance on the artery and cause it to dilate to open up. So maybe a little carbon dioxide, maybe a little nitrous oxide, a little NL. So why are we monitoring filtrate osmolarity? Because remember part of the job of renin, well, indirectly. Renin is going to set up that whole system of change where we get angiotensin II. Angiotensin.
Bro, this weekend I want to make like the Dubai chocolate cookies. Yeah, they're gonna be so fun. It's gonna be so fire. Yeah, sure. If I have them. Oh, you're right. You're right. My bad. Look it up. Look it up on Instagram. Look it up on Instagram. Look it up on Instagram. Okay, I'll show you. I'll just show you. Can you see can you see my screen? This girl, where did her waist go? Are you saying waist for her? She's pretty. Oh, her. Oh, her. Yeah. I have more workout videos which I'm on TikTok. Like, mm, mm. Bro, I need to make this. It looks so fire. It's cauliflower, bro. This. It's literally cauliflower. It looks so fire. be differently.
But I followed her thing, Instagram thing. What's it say? Vegan cake. Oh my god.
I thought it was for June.
Wait, yeah. Wait, why? How do you follow all these people?
Most of these people are from my church.
Oh yeah, I was like, oh, how? A lot of these stories were just like celebrities.
Awesome.
Most of that's God, where are you going? Oh my god, where are you?
What?
You go so fast.
I'm an expert.
Oh my gosh. You actually slide through every person. Like I don't. I just look at two and I'm like, yeah, that's.
Sometimes. So cute. Look, it's you. Okay. Let me. Oh, shoot.
Alright. Yeah, I had beef. Thanks.
Two is going to stimulate the release of aldosterone, and aldosterone is going to cause an increase in sodium reabsorption at the distal tubule. So if filtrate osmolarity is falling, if the fluid inside the tubule is becoming more dilute, it suggests that the renin system is already in play. And we've already got the systolic blood pressure, so the distal tubule says, whoa, the fluid inside me is becoming less concentrated quick. Stimulate the release of renin and open up that artery. Renin goes through its whole big thing, converts everything to angiotensin II. Angiotensin II comes and constricts all of the arteries in the body, including both of these guys. And then the distal tubule comes back and opens up selectively, just the afferent arteriole. Now we have that more blood in, less blood out scenario. We're going to capture volume inside this capillary, then an increased pressure. We've got a big systemic increase in pressure, so we're driving more blood to the kidney in general. Now we fixed our general systemic pressure issues, and we fixed our local pressure issues. So we're going to have a nice perfusion throughout the body. We're going to have a good increase in filtration rate. Now, if blood pressure is too high, I have to be able to balance both low blood pressure and high blood pressure. If blood pressure is too high, this is going to be sensed by the right atrium of the heart. The atrium stretches out if blood pressure is high, and it's going to release a substance called ANP. ANP stands for atrial natriuretic factor, ANP, or atrial natriuretic peptide. Either way, ANF or ANP. And ANP basically reverses everything that angiotensin II does. It's going to act as a vasodilator to open up blood vessels and reduce blood pressure. It's going to inhibit the release of renin, so it's going to shut down that system that's driving blood pressure up. And it's going to inhibit the release of aldosterone. So again, preventing psyllium from being retained, and thus water, and that's keeping blood volume low. Let's summarize what we talked about. Not formalism. Large fat blob, little fat blobbies like this. Let's say you were smashing some ground meat into hamburgers and you've got 22% ground meat fat on your hand. And you go over to the sink and you turn on the water and try and wash it off. And nothing. The water just rolls right away. It doesn't take the fat with it. So you take soap and you rub some soap between your hands. And soap does this to the fat. Now you hit it with the water, and the water to grab this little polar shell and whoops that right off your hand. And it's the same basic idea here. To break up that fat into smaller globs, allow it to mix into the system better, and allows it to be more easily digested. Bile is made by the liver and stored and concentrated in the gallbladder. So the gallbladder produces a nice, concentrated squirt of bile that can get dumped into the intestine whenever you eat a high-fat meal. So important to note that bile does not digest anything. Bile is not an enzyme. Bile just breaks up the fat so it's easier for the lipase, the enzymes, to get in there and digest it. Sometimes the gallbladder does its job too well, and the bile starts crystallizing. And when the bile starts crystallizing, then you start to get bigger crystals, and sometimes they mix up with cholesterol or whatever else, and they jam up the duct that leads out of the gallbladder. And then that causes the gallbladder to swell up and become inflamed and painful. Everybody has a few of these little bile crystals. We call them gallstones. But if the gallstones get too big and really start impairing gallbladder function, then they'll just go in and take out the gallbladder. You can live without your gallbladder. You still have the liver making little wimpy kind of bile here, but you can't live without your liver. And if you live without your gallbladder, it just means you can't eat really high-fat meals anymore, because it would be very uncomfortable to do so. If you don't have that nice concentrated bile, then some of that fat's going to remain undigested. And when it gets to the large intestine, bacteria will break it down. And that can lead to bloating and cramping and general uncomfortable.
Okay, I'll do it right now.
Indigestion kind of feelings. The third digestive accessory organ we're going to talk about here is the pancreas. Now, the pancreas has a dual role. It is both an endocrine organ and an exocrine organ. So it produces hormones. The two big hormones coming out of the pancreas are insulin and glucagon. And these, of course, are involved in carbohydrate regulation, blood sugar regulation. The exocrine role of the pancreas is pretty significant. This guy is a major source of digestive enzymes for your GI tract. It produces at least one and sometimes several enzymes for each of the major macromolecules. So several proteases, a couple of different lipases, amylase to digest starch, and nucleases to break down nucleic acids. And in fact, people who have pancreatic problems like pancreatitis or pancreatic cancer, often the side effect of those illnesses is malnutrition, because without the pancreas and without the digestive enzymes it supplies, it's very difficult to break down your food and absorb the nutrients. Now, the other exocrine product that the pancreas is bicarbonate, because all of these enzymes are getting dumped into the small intestine, and most enzymes prefer to work at a neutral pH around 6, 7, 8. But the stuff coming out of the stomach into the small intestine is at a very low pH. Stomach is very acidic, about pH 1 or 2. So in order to neutralize that acid, and bring the intestinal pH back up to a more neutral level, the pancreas is going to release bicarbonate along with all of these digestive enzymes. That helps to bring the intestinal pH up to around 6 or 7, which is a much better optimal pH for these enzymes to be functioning at. Let's summarize what we talked about in this video. We looked at the difference between the alimentary
Jesus, I praise you. I praise you and I worship you, God. Your mercy. God, make it alright. Only you can make it alright. Dear God, make it alright. Only you can make it alright. Alright. Only you can make it alright. Alright. Dear God, make it alright. Only you can make it alright. Dear God, make it alright. Only you can make it alright. Good evening, cat.
In this video, DNA repair mechanisms, in particular, focusing on end joining and direct reversal. Mutations in the DNA can be caused by a number of different things, such as polymerase errors, which leads to point mutations, repeats, and small insertions and deletions. Endogenous damage, which can lead to oxidized bases or physical damage to the DNA. Exogenous damage, such as radiation and chemicals, can lead to dimers and also break DNA. Or transposons, which can lead to large insertions and deletions, inversions or duplications. Generally speaking, transposons do not lead to repair mechanisms because there's no visible damage to the DNA. The DNA has been moved around, some taken in, some taken out, but there's no visible damage, so they don't trigger repair mechanisms. Mismatch repair pathway, nucleotide, excision repair, focuses on a single base that's been either mispaired during replication or is damaged somehow. So here you're just going to fix the mismatched base, and here you're going to cut out the old base and put in a new base. So nucleotide excision repair and mismatch repair is used to fix polymerase errors. It can also be used to fix damaged bases due to either oxidation or chemicals. If the DNA is physically broken, then we have to do some end joining. And there's two possibilities for this: homologous end joining. So let's take a look at homologous end joining first. Homologous end joining, as let's spoken, then we have to do some end joining. And there's two end joining. So let's take a look at homologous end joining first. Homologous end joining, as we said, is going to repair double-stranded DNA breaks. This particular type of repair has to happen after replication. It requires the use of a sister chromatid as a template to repair the broken strand. So here you can see a pair of sister chromatids after replication. You can imagine up here there's a centromere, and the rest of the two chromosomes are here. And so one of them here has some damage, a broken strand. So what we can do is first do a little cleanup on this broken strand. Get in there with some endonucleases and exonucleases to clean up the damage. Then we're going to use our intact sister chromatid that can cross up over here and base pair with the busted strand. And we can put the busted strand down here to base pair with the sister chromatid, so it ends up looking like this. And now we can use these two strands to replicate each other. We can use this template on the bottom here to replicate the blue strand. We can use the pink strand on the top of the template to replicate the blue strand here. Now, this is the best way to repair double-stranded breaks, because you're using an existing good piece of DNA to repair the bad piece of DNA. But if this is not possible, maybe you're not doing DNA replication because the cell's not going through cell division, so you don't have a sister chromatid to use as a homologous partner to repair the problem. So non-homologous end joining pretty much just sticks the two pieces of DNA together. Here's a little busted piece of DNA. As usual, we're going to go in and do some cleanup, and then we're just going to join the two ends together. And this type of repair is almost always mutagenic, because you're going to be losing some bases in that busted DNA strand. Plus, worse, you could connect the wrong part of the chromosome to the wrong part of another chromosome, resulting in some translocations. We're going to look at a picture of that in just a second. But the idea is that this is better than having a busted DNA strand. It's better than nothing. It's mutagenic, but it's better than nothing. So in a translocation, let's say you had two chromosomes that had double-stranded breaks. You just accidentally stick the wrong chromosomes together. So this little piece of chromosome 20 gets stuck onto chromosome 4, and the bigger piece of chromosome 4 gets stuck onto chromosome 20. This is the type of error that can occur with non-homologous end joining. Worse, if this particular joining point is in the middle of the gene, it can lead to gene fusion products. If you do a little inversion here, little translocation, you could pair the wrong part of one gene to the wrong part of the other gene and get fused gene products. The second type of repair that can happen here, homologous and non-homologous enjoining can happen here if you've got physical breaks in the DNA. It can also happen here for breaks caused by X-rays or chemicals. The dimers that are caused by UV radiation can often be reversed by simply exposing the DNA to visible light. White light reverses these pyrimidine dimers. So here we have a piece of DNA. White light reverses these pyrimidine dimers. So here we have a piece of DNA, and it's been exposed to UV radiation, and that produced a couple of pyrimidine dimers. If we exposed the DNA now to plain old white light, it simply reverses the dimer. There's no extensive, complicated pathway. It's just a direct reversal. Let's summarize what we talked about in this video. We briefly discussed the DNA repair mechanisms caused by different mutations, and in specific, we focused on homologous and non-homologous end joining, as well as a direct reversal of dimers caused by UV radiation.
Be back.
Probably gonna go to sleep. I'm probably gonna go to sleep. Night.
we'll be discussing the differences between prokaryotic and eukaryotic transcription. There are a fair number of differences between prokaryotes and eukaryotes, and most of them are in molecular biology. They have different replication strategies, different transcription strategies, different translation issues. So we're going to be focusing on the differences between transcription and a little bit of translation stuff. For example, in prokaryotes, transcription and translation happen at the same time and in the same place. Both of them are happening in the cytosol. Transcription in the cytosol, translation in the cytosol. As the mRNA is coming off of the RNA polymerase, ribosomes are binding to it and translating proteins. For eukaryotes, not so much. First of all, the transcription and translation happen in different places in the eukaryote. Transcription happens in the nucleus, while translation is happening in the cytosol, so that it can't be occurring at the same time. Besides, even if they were happening in the same place, the mRNA that comes off the RNA polymerase initially is not mature mRNA. It's officially called hnRNA, which stands for heterogeneous nuclear RNA. And this is RNA that is unprocessed. So before that can be translated, it has to go through mRNA processing. This includes adding that 5' guanine cap, a 3' poly A tail, and splicing to remove the introns and splice back together the exons. Once that's ready, the mature mRNA is moved out to the cytosol and then translated. Prokaryotes don't do any sort of mRNA processing. As I mentioned, as soon as the mRNA comes off the RNA polymerase, it's ready to be translated. Prokaryotes are polycistronic. This means that from a single piece of mRNA, you can get several different proteins. They can use different reading frames to get different amino acid sequences. They can use nested genes. They can have 3 coding regions in a row. So you can get multiple proteins off a single piece of mRNA. Eukaryotes are monocistronic. This means that they've pretty much got 1 protein per mRNA. Once the mRNA is mature and out in the cytosol, you can't get different versions from it. You might have heard this as one gene, one protein, but that's not quite accurate, because a single gene can be spliced in alternate ways to produce slightly different mRNAs, and thus slightly different proteins. But once you have that mature mRNA, you're only going to get one type of protein from it. Lastly, prokaryotes use a single RNA polymerase. They have 3 or more different DNA polymerase, but they only have a single RNA polymerases. While eukaryotes have 3 different RNA polymerases, RNA Pol1, Pol2, and Pol3. Pol1 transcribes rRNA, Pol2 transcribes mRNA, and Pol3 transcribes tRNA. And you can remember this, because these names, Pol1, Pol2, and Pol3, are pretty much meaningless. They are empty. Pol-I for rRNA, Pol2 for mRNA, and Pol3 for tRNA. Let's summarize what we covered in this video. We discussed the differences between prokaryotic transcription and eukaryotic Pol2 and Pol3. Pol1 they have 3 or more a single races. 3. Pol1 transcribes races. RNA Pol1 races. While eukaryotes have 3 different RNA, you're only different spliced 1 protein per mRNA are monocistronic. This means that they've pretty much got 1 protein per mRNA. Once the mRNA is mature and out in the cytosol, you can't get different versions from it. You might have heard this as one gene, one protein, but that's not quite accurate, because a single gene can be spliced in alternate ways to produce slightly different mRNAs, and thus slightly different proteins. But once you have that mature mRNA, you're only going to get one type of protein from it. Lastly, prokaryotes use a single RNA polymerase. They have 3 or more different DNA polymerase, but they only have a single RNA polymerases. While eukaryotes have 3 different RNA polymerases, RNA Pol1, Pol2, and Pol3. Pol1 transcribes rRNA, Pol2 transcribes mRNA, and Pol3 transcribes tRNA. And you can remember this, because these names, Pol1, Pol2, and Pol3, are pretty much meaningless. They are empty. Pol-I for rRNA, Pol2 for mRNA, and Pol3 for tRNA. Let's summarize what we covered in this video. We discussed the differences between prokaryotic transcription .
17-minute video.
Structures and protein transfer. These are RNA. Remember that the enzyme that creates the primer for DNA replication is an RNA polymerase, and the primer is a little piece of RNA. So RNA polymerases can start a strand from scratch. So we don't need a primer for transcription. And lastly, there's no editing in transcription, no proofreading. And you think at first, well, that's bad, because if we don't edit our transcript, if we don't proofread, then we're going to end up with some bad RNA. And that's true. You will end up with some bad RNA. Whether or not you get bad protein is a different story, though. For one thing, we don't worry about it too much, because even if it was completely bad RNA and produced completely bad protein, it's a transient molecule. It's going to be degraded and made again, and the next time you make it, it'll probably be fine. Between the promoting, you're transcribed using a strong promoter. A weak promoter has a lower affinity for RNA polymerase, so RNA polymerase will bind and stick sometimes, and sometimes it will bind and fall off. So if it binds and falls off, obviously you're not going to get any RNA. So you're going to end up with less RNA from a weak promoter. The problem with this system is that you can't really change this. What you've is just a sequence within the DNA. So if you suddenly need more of a particular protein and you have a weak promoter, you can't swap it out for a high promoter, a strong promoter, and then get a lot of mRNA for that protein, and thus a lot. We're going to be discussing it here. Rho. So for can bind to the repressor, and when lactose, the poly-A tail in eukaryotes is not a string of T's in the DNA. It's made by a completely separate enzyme called polyadenylase. So the stop site in eukaryotes is the polyadenylase signal. In prokaryotes, it's a different sequence, because prokaryotes don't do polyadenylation. But there's still a...
Quick reflection. Start earlier. Start earlier, and I also want to do the question banks. I have those question banks. Why am I not doing them? Need more time. It's annoying. Let me just go sleep for